back one. The theory is called the fluid mosaic model. Fluid mosaic model. What does it mean? That is, it tries to explain the position of proteins in relation to the lipid bilayer. So in the electron micrographs, we can see two black lines and one white ones. So based on this, people propose that the black ones are proteins, and the center ones are the lipid. So like a sandwich model. That is 2 layers of proteins plus 1 layer of lipid. But this is not consistent with the physiological studies. The physiological studies thinks that there are proteins which are located on different sides of the membrane. That is, you can see, those are on the surface. Those are striding across the plasma membrane. And so scientists gave them different names. One is called integral. Integral means stuck into as an integral part, as a unifying part of the plasma membrane. It's called the integral protein. So these are not likely to be washed by salt. However, there are some proteins which are easily washed out by mild treatment of salt. So this is called the peripheral proteins. Okay, that is, it's near the surface, or it's attached to the surface, where its attachment to the membrane is not so tight. Okay, so based on proteins which have different affinities to the phospholipid bilayer, then this person, I think it's in the 1970s, proposed this fluid mosaic model. Fluid means that this lipid bilayer is a fluid, is a 2-dimensional fluid that it can flow. And these lipids can flow on this 2-dimensional surface. It's called fluid. Mosaic means that these are the mosaic inserted into the membrane. So these proteins serve as a mosaic on the sea of the plasma membrane. So this is proposed by Singer and Nicholson in the 1970s. And this theory was receiving more support in the past years. And so this theory was generally accepted. Of course, there are more additions to this model. That is, it has some lipid raft. Raft means a small boat, right? The scientists discovered a patch of lipid which on the lipid surface as a raft, as a small aggregate. It's called the lipid raft. And the general theory is this. Phospholipid can rotate and move laterally. So these are very liquid. And these are floating in the liquid. And so the whole phospholipid is viscous as the olive oil. That is like a liquid. However, this liquid is quite sturdy. So it can prevent things from getting in and getting out. So this is very important because the cells must maintain a boundary for the inside and for outside. If there's no boundary, then its inside comes out, and then it's dead. Okay, so plasma membrane serves many functions, and these functions are very important for the living of the bacteria. So I'm proposing the 3 main functions to explain. First is selective permeability. Second is energy. And the third is the photosynthesis. That is for the photosynthesis to work on the lipid of the chromatophores, the plastid or the thylakoid of the chloroplast. Okay, let's first explain the permeability barrier. So it's simple, because a cell as a living thing must maintain inside and outside. So inside material cannot leak out if it wants to keep it, and the outside bad things cannot come in if it wants to keep its life. So this permeability barrier is very important. And how to do this? Because this is a lipid. This is a lipid. So lipid soluble molecules generally can get in and out easily, like ethanol, as small as water. Water is not purely organic, so it's in and out of the cell with some difficulty. For the ions, that is the charged particles, it's a no-no, because the lipid doesn't like the ions, the charged molecules. For large molecules, it's also a no-no. So the lipid bilayer generally is a gate for the aqueous, not water-soluble molecules to get in and get out. But it does need to have to take some nutrient in and get some waste out. So it has to develop a certain kind of transport. So let me explain the presence of the transport. First, for the molecules to move across the membrane, simple diffusion. Simple diffusion means that it's able to get into the inside just because of diffusion from high concentration to low concentrations. It's called simple diffusion. The simple diffusion is really slow. Even for water, it's very slow. Not to mention ions, large molecules. So it has to have a facilitative diffusion. Facilitative diffusion means that there must be a transporter, that is to grab the molecule and take it from outside to inside, or grab a molecule from inside to outside. So these are the important evolutionary product of the bacteria, is that it has to have a transporter as both peripheral proteins and integral proteins, and which is able to facilitate the in and out transport of molecules. Let me show you the three basic systems. A simple transport, that is energy coupled with the transport protein can transport one molecule inside the cell from outside to the inside. So this is called a simple transport. Simple transport must have energy to help it. This energy generally is the hydrogen potential, hydrogen ion potential. That is electromotive force to drive the passage of this substance. So it's a simple transport. This molecule is transported intact, that is unmodified. However, group translocation is another system. That is this substance, when it's imported, then it was modified. This system is called group translocation. Group translocation requires a transport code. Group translocation. So this is very important because glucose, such as lactose, these kind of molecules, when it was transported, it has to use one energy molecule, so PEP, phosphoenolpyruvate, PEP. This is the molecule in the glycolysis pathway. It has a phosphate, phosphate, the bond. This high energy phosphate will be transferred during this process to the sugar molecule. So this sugar molecule is phosphorylated. And the interesting thing is that this is modified, so that once it is phosphorylated, it will be metabolized. It will be metabolized through the glycolysis. So you cannot get out again because it is phosphorylated. So it's more difficult to get out. So this process is called the group translocation. E. coli has a few of this. It's system called the phosphotransferase system. So it can transfer various kinds of group sugar from outside to the inside, and at the same time phosphorylated. So glucose become glucose 6-phosphate. Okay, and this is a system we can see the many proteins. This is number 1 protein, number 2, number 3. And it's called the ABC system. ABC is called the ATP binding cassette. ATP binding cassette. ATP binding. Where is the ATP binding? This one. This is a small cassette. So this cassette provides the energy for this protein, transport protein, major integral protein, to bind this peripheral protein, that's outside protein. The outside protein binds a substrate. It will transfer to number 2, the integral protein. Integral protein will change its conformation. And with the energy provided by this number 3, ABC ATP binding cassette, and so as to get the molecule into the cell. So it is the ABC system. This ABC system extensively exists in eukaryotic cells, hundreds of it, and in prokaryote cells, also hundreds of it. So it means that this ABC system is widespread and is very important for both prokaryotic cells and eukaryotic cells. So this is a simple introduction of the membrane transport system. So this is a very important part in the metabolism of the cell. Okay, let's go back to the another functions of another function. That's it. And is a protein anchor is explained, and then the energy conservation. This is also important because cells need to have energy store. This energy store is in the form of proton potential, proton motive pump, proton potential. This proton potential is, that is, the inside is, the outside is more hydrogen ions. The inside has more hydroxide ions. And this is positive, that is, this is positive, more positive, and the inside is more negative. So it created electrochemical gradient. And this gradient, when hydrogen ion comes in, if with a protein molecule, generally called the ATPase, ATP generating machinery, then this protomotive force is able to generate ATP or do some work. So this process called the energy conservation. Bacteria uses its plasma membrane as the energy conservation site. For the prokaryotic cells, this structure is the mitochondria. That is, prokaryotic cells, eukaryotic cells use the mitochondria, which is originally an independent microbes to do the energy conservation, energy preparation. and also for the energy production, that is ATP production. So I have mentioned that the three main functions of the plasma membrane. That is the transport barrier first, and then transport, and then the third is energy conservation. Okay, so let's have another 10 minutes rest for you, and let me start here. So you can take a little rest for the time being.